Yes, so today we are going to discuss the next part of this chapter, that is principle of inheritance and variation, law of dominance. Now, this was being said by Gregor Johann Mendel. What did he say in this law? He said every gene, now look at it very carefully, every gene contains information to express a particular trait. Now, what does this mean? Every gene contains information to express a particular trait. Yes. Take for example, if I write down capital R and capital R, this suppose represents rate, dominant rate. So this is what this law actually tries to tell us. Now, one thing that we need to remember over here is, suppose we are taking an example of gene producing an enzyme. Let us assume, as we know that a gene, a group of genes comprises together to form a mRNA, and the mRNA changes itself to a protein, and the protein represents a particular enzyme which carries out various functions in our body. Assumes normal allele produce normal enzyme required for the transformation of substrate is. Now understand this thing. I always believe that the NCERT has given a thorough explanation of this topic. We will be explaining, or we will be understanding what it tries to tell. A gene produces an enzyme. We all, as I said. Now let us assume a normal allele like capital R, small r, or a capital R, or capital T, small t, produces normal enzyme, which is required for the transformation of a substrate. As we all know that any product, any reactant changes into a substrate. How? By the help of the enzyme. Now, based on this, let us talk about two types of alleles can be formed. One can be a modified allele, other can be a unmodified allele. Modified allele, what does this mean? Modified allele means, or rather the unmodified. Unmodified means whatever the allele it was having, suppose capital R and capital R, after the enzyme, means when the reactant was capital R and capital R, after the enzyme has reacted onto this reactant, it has changed to the substrate. At that time also its alleles are same. There is no change. That is what we are calling as unmodified allele. Unmodified allele means normal allele produce normal enzyme needed for transformation of substrate. Whereas a modified one means, suppose over here the reactant or whatever the substrate it was, it has changed into a product. Reactant or the substrate, we call it in the according to the biological term. If now this was capital R and now it has changed to capital R and small r, now this is a modified allele. The modified allele can be of either it can be a normal or less efficient enzyme. It can be a non-functional enzyme, it can be no enzyme produced. So what does this mean? That after modification of the substrate to the product by the help of the enzyme, what I have seen that the alleles have transformed, it has changed. So if it has changed, then what are the three possibilities? These are the three possibilities. Like normal allele or normal efficient, less efficient enzyme is equivalent to normal allele to produce same phenotype, means whatever the thing I had got in the beginning, after it has been modified, it remains almost same or nearly the same. Means the phenotype that I will be getting, it will be the same. There is no change in that. And non-functional enzyme or no enzyme produced is, it depends upon the phenotype. It depends upon the phenotype. Means phenotype depends on normal allele. This is the thing, normal allele. Non-functional means, suppose just think of an example. Out of the two alleles, capital R and small r, this one is a dominant one. Now suppose this allele is unable to function at that time, then which one will be functional at that time? This small r. So that is what we are calling as non-functional enzyme, or we are calling it as a recessive one. Recessive one means at that time which one will be representing the phenotype? This small r, like capital R and capital R, small r represents pink, deep pink. Now if suppose this one is non-functional, then which one will be representing its phenotype? The small r. So small r over here is the recessive one. So this is what we are talking about, non-functional enzyme or no enzyme produced. So remember that every gene contains the information to express a particular trait. Trait means character. And this is why I have given you this example, that alleles can be modified or unmodified. Unmodified means it will be remaining the same. There will be no change in the allele formation. Means whatever the enzyme that I had got in the beginning for the transformation of the substrate, it remains the same. Like capital R and capital R was the allele which after transformation it remains the same, capital R and capital R. Whereas if it has become modified, it can become either the normal as the same one that we had got in the beginning, that is capital R and capital R, or less efficient, that is capital R and small r, which is equivalent to the normal allele that produces the same phenotype or nearby the same phenotype almost. And other than that, it can also become non-functional enzyme. Non-functional means out of that two alleles, one becomes non-functional. Then what will happen? The small r will be functional. Means suppose I am a right-handed person. If my right hand somehow gets damaged, then I will be working with my left hand. So that is what I am talking over here. That is the non-functional enzyme. That is the recessive one, which we call. Dominant is this one, this hand. When I am not able to work with my the right hand, then my recessive hand is the left hand. So that will be the functional at that time. So this is what law of dominance is. Next one is law of or rather not the law, we are going to now talk about the incomplete dominance. We are now going to talk on the incomplete dominance. What is incomplete dominance? Always try to relate anything, because see, after studying everything, we always, before the exam, we always forget that thing. So try to relate that with the words or the topics that you were studying. allelic interaction or intragenic interaction. Now this term, under this incomplete dominance comes. Allelic interaction means within a gene. Suppose there is capital R and small r, as I say. Now over here, as you can see, these are the two alleles. They will be interacting among themselves within a single gene. That is what we call as allelic interaction, or intragenic means within a gene when two alleles are interacting with each other. That is known as the intragenic interaction. Now over here in this, dominant factor is unable to express its character completely, results in formation of intermediate type of gametes. Or intermediate type of generation, which differs from both of its parents. Now this is what it is being stated, but this does not mean this is not blending. Why? Because parental types are recovered in F2 generation. Now let us take an example of Mirabilis jalapa or 4 o'clock plant. I have taken capital R, small r, small r. This is red, this is white. Now from here one allele comes, from here one allele comes, and together it forms capital R and small r. These are the gametes, and this is pink in color. Now if I do the self crossing over here, what it will be? Capital R into smaller across, getting the cross. So what it will be? So as you can see, I had started with capital R, small and small r. So in the F2 generation, what I am getting over here, I am again getting back the parental types. So that's why I said that this does not mean blending. Blending would not have given me this. It would have always given me a mixture. So that's why when in the F2 generation you are getting back the parental types, that means it is not totally a blending. It is what we are calling as incomplete dominance. This capital R, if I write down the genotype, of this one, it will be 1:2:1, and the phenotype will also be 1:2:1. As you can see, one is a pure dominant, one is a pure recessive, and the two other ones are the hybrid ones, which give a pink color. It's the intermediate one, which represents or which differs from both of its parents. Phenotype will also be the same thing. So genotype and phenotype in incomplete dominance will be of same type. It is not the different one. Now I am going to discuss on codominance. What is codominance? In codominance, in heterozygous condition, both the alleles are expressed. Means here both the alleles will be expressed. Both alleles are dominant. The characters are intermediate and in between both the traits. Remember, in incomplete dominance, I say that the traits are intermediate, but they are not in between both the traits of the parents. Fourth point is, in F1 generation resembles the parents. Means in F1 generation only the parental generation will be resembling. Now let us take an example. Like for example, AB blood group. Over here you will be finding that both A and B antigens are equally present on the surface of the RBC, means both are equally expressed. So this is one of the best examples of AB blood group. Another one is sickle cell anemia. I will be discussing with this. As in sickle cell anemia, what you will be finding in sickle cell anemia, this is hemoglobin A, this is also hemoglobin A. This is a normal blood cell, these are the blood cells. In the carrier, what you will be finding, in the carrier you will be finding this is also hemoglobin S, this is hemoglobin A. This is a carrier and these are the blood cells. In the affected individual, what you will be finding, in the affected individual you will be finding hemoglobin S, and over here also you will be finding hemoglobin S. This is an affected one. So what does it mean? In the normal blood cells you will be finding that in us hemoglobin, HB means hemoglobin, and A is the normal type of hemoglobin that is present in our blood cells. Now suppose a person is carrying this disease, that is sickle cell anemia. What is sickle cell anemia? Out of the two hemoglobins over here, that is A and A, in the carrier you will be finding there is a different type of hemoglobin, that is hemoglobin S. It's having a sickle shaped. It's a special type of shape which we call as sickle shaped. So over here you will be finding that in the carrier both A and both S, both are equally dominant over here. No one is recessive or no one is totally dominant. But when I am talking about the affected one, what happens in the affected one, only hemoglobin S is being present over there. That means this person will be dying within a very short period of time. Hemoglobin of normal us, it has a lifespan of 6 months, but whereas they have a 120 days, whereas this hemoglobin S has a lifespan of 30 to 40 days only. Now, as I was talking about codominance, over here you'll be finding only both A and S are equally involved in the carrier persons or in the carrier blood group. That is, both are having equal importance. This is what sickle cell anemia is all about. Now, I will be talking about another thing, that is about the blood group. In the blood group, what you will be finding, and in the this is the genotype, you will be finding there is a I gene, and it has got three alleles A, B, O. So when the genotype is I, B, I, O, or I, B, and IB, it is having such type of blood group, means its antigen. Or when the blood group is IA and IO, in the AB blood group, you will be finding the combination can be IAB or IBA. And in the last one, where there is no antigen, you will be finding that in the I gene there is no such A or B antigen present. A and B are dominant ones, and O is the recessive one. This is what it is being stated over here. Now, as I said, we are going to discuss only, because I will be giving our next video where you will be finding in this chapter only we will be studying blood group in much details. I will be explaining you. Right now, see, suppose I am giving you an example. A mother whose blood group is, or who is having IA and IO, and a father who is having IB and IO. When they are having a cross, what do the first child will be having a blood group? IA and IB. After that, what it will be? IA and IO. Again, it will be IB and IO. So these are the four possible cases. But see over here, both the genes A and B are equally responsible for the blood group AB. That means on the surface of the RBC, both the antigens are equally participating and both are dominant over here. It is not like that one will be able to express more or one will be more pronounced than the other. Like in incomplete dominance, I said that the dominant gene will be able to express, but the recessive one will also express. Means in incomplete dominance, the dominant nature of one gene was not totally expressed. But over here in codominance means both the genes, co means both are together and both are equally involved in the genetic constitution of that particular organism or particular gamete or the particular phenotype also. This is what codominance is about. Now today's last topic is multiple allele. Multiple allele. What is multiple allele? First of all, remember, more than two alternative form of a gene located on the same locus of homologous chromosome. Locus, the better term of locus, you can read it as location. Third point is reason is due to mutation, that is due to genetic error. Characteristics. What are the characteristics? Number one is a diploid individual contains two allele, and each gamete contain one allele for a character. Yes, we all know that the two alleles from the parental generation, they do get separate and each gamete gets one allele. So that is what it is being said. Second characteristic is they show meristeric type of germinal variation. Meristeric means from parents it passes to offsprings, and from offsprings it goes on. It's a particular type of a character or a trait. That is, due to the evolution, you will be finding that continuous variation and discontinuous variation, that is from one generation to the next generation, when a particular type of allele goes on or a particular type of character goes on. That is what the word meristic comes from. Germinal means, you can understand that from the parents or from the parental type of alleles, the characteristics that a particular offspring bears, and when it passes it to the next generation. That is what we are seeing. Next is all multiple alleles occur on same gene locus of same or homologous chromosome. And now comes the fourth point, that is the last point. There may be many alleles in a population alleles of a gene in a population, but in diploid individual only two alleles of a gene are present on same locus of homologous chromosome. So you must be thinking that such a long statement sir is written on the board, so don't worry, I am explaining it to you, just listen. Now see, more than two alternative forms of a gene. Yes, till now I have said tall. These are the possibilities, means two alleles together represent a particular type of allele, that is tall. Now suppose blood group. How many alleles a blood group has got? I gene has got 3 alleles. So it means a single gene, when it has got more than 2 alleles, we call it to be as a multiple allele. And located on the same locus of homologous chromosome means, locus means, this word means location, on the same position of 2 homologous chromosomes. Reason is due to mutation, as I said, it's due to a change. Now the thing is, a diploid individual contains 2 alleles and gamete contains 1 allele from a character. As I said, that this is a allele coming from a parent, from your father, and this is a allele coming from your mother. So 2 alleles will be going to 2 different gametes. It will be making 1 gamete and it will be making 1 gamete. So that is what it is being said. Like, this is a chromosome and this is an allele. Now this is from your father and this is from your mother. Now when there is a replication, what will happen? This one will be having a homologous chromosome and this one will also be having a homologous chromosome. Means after replication, each one forms a homologous chromosome. Father has got 1 homologous pair of chromosome and mother has also got 1 pair of homologous chromosomes. This is what we try to say. Meristic type of germinal variation means, as I said, that from your parents, when one particular trait passes on to the next offspring and from that to the other, that is what we call as meristic type of germinal variation. All multiple allele occur on same gene locus of same or homologous chromosome. Yes, this one is a little bit what I need to say. Suppose this is a chromosome where you are finding this is P, this is A. Similarly, this is, here is also P, here is also A. Now this is the chromosome of a person said Ram. Now there is another chromosome which is having, and this person's name is Sam. Now you can see that both the position of the alleles are having same distance. P is present over here, P is present over here, and see P is also present over here. So the distance between the alleles of Ram and Sham are of equal length. That is what we are calling same gene locus, means in the same position of the homologous chromosome. Homologous means these two are homologous, these two are homologous. Now see, over here in Sam also the distance is same, over here in Ram also the distance is same. So that is what we are calling as homologous chromosome. The alleles are of same length. Now there may be many alleles of a gene population. Now suppose in Durgapur there are many alleles. Plenty of alleles are present in many of the individuals. But in a diploid individual only 2 alleles will be present. Like over here you can see in Ram, what are the 2 alleles? P and A. In Sam, what are the 2 alleles? P and B. So are the 2 alleles, suppose, so how many alleles are there? P, A and B. So in a diploid organism, always there will be 2 alleles. It is not like that the alleles will be same. It may be different also. You can find in some other person of Durgapur the 2 alleles A and B are present, maybe B not be present. But as it is being said that are present on same locus of homologous chromosome, same locus C, the distance between P and the distance of P in Sam. Both the alleles are having same distance of the homologous chromosomes. So this is what I tried to say, that what is multiple allele, a single gene, maybe a homologous chromosome will be having only 2 alleles. But a particular gene can have plenty of alleles, but in a particular individual only 2 alleles of that particular gene will be present. This is what multiple allele is all about. Thank you. If you are having any queries, you can ask it to me and you can subscribe my YouTube video. Thank you.